Welcome to LankanTutor Biology 2.6, and today we're going to be talking about nucleic acids. Most of you already know about the DNA and the RNA. It was a molecule which was found by James Watson and Francis Crick, who figured out that the genetic materials of your cells get stored in this wound up structure called DNA. Different cells have different genetic material. For example, we have DNA, and there are plants that have RNA, and so we'll get into the microscopic structure. If you do want to learn a bit extra about DNA, there is a video done by Crick himself on a YouTube channel called TED Talks. You might want to Google that. Just Google TED Talks, Watson and Crick. You should get a brilliant video if you are interested to learn more. They talk about how they actually found the molecule and everything. It's really interesting. So let's get into the stuff that we actually need to study. So we're learning about nucleic acids. A nucleic acid is a macromolecule, and that means that it has a molecular weight of 10 to the power 4 to 10 to the power 10. So it has a pretty high molecular weight, and it's a linear polymer. It is not branched. It comprises of small repeating units, like all polymers, and those units are called nucleotides. A nucleic acid contains carbon, hydrogen, oxygen, nitrogen, and phosphorus. So this is the final organic molecule we're studying about. Now, if you remember carbohydrates, it was carbon, hydrogen, oxygen, and in lipids, it was carbon, hydrogen, oxygen, nitrogen, or phosphorus. Now, the nucleic acids have three components. One is a pentose sugar. A pentose sugar is a monosaccharide which has 5 carbons. It has a nitrogen-containing base and a phosphate group or a phosphoric acid. So remember that. Those are the three parts of a nucleic acid: a pentose sugar, a nitrogen base, and a phosphate group. There are two types of nucleic acids, and we all know about these ones. It's DNA and RNA. DNA means deoxyribonucleic acid, and RNA means ribonucleic acid. The difference between DNA and RNA is the fact that the two different nucleic acids contain two different types of bases. The bases that go into these molecules are adenine, guanine, uracil, cytosine, and thymine. We can divide this into two groups called purines and pyrimidines. Adenine, guanine are purines, uracil, cytosine, and thymine are pyrimidines. What you need to remember about this is that DNA contains adenine, thymine, guanine, and cytosine, which is ATGC, and RNA, instead of thymine, it contains uracil. That would mean adenine, uracil, guanine, and cytosine. The thing is, the DNA molecule, which we're going to go into next, is a double-stranded structure, and a purine always connects with a countering pyrimidine. We'll get into that soon. So this is how the DNA structure works. You can see it has a pentose sugar, and each pentose sugar is connected to a phosphate group and a nitrogenous base. It could be adenine, guanine, thymine, or cytosine. The thing about the DNA structure you need to remember is that they are counter-parallel or anti-parallel. So that means it has two nucleotide strands which are facing the opposite direction, and the purines of one strand connect to the pyrimidines of the other. Adenine can only be bonded with a thymine with an adenine. A guanine can only be bonded with a cytosine, and vice versa. So there you have it. That is how a DNA molecule looks like. And when this entire thing gets elongated and it curls up and up and up and up and up, it's what makes chromosomes. We'll get more into that in cell division, but generally the DNA strand has a sequence of bases which stores genetic information, and that is what you need to know. So these bonds between the nucleotides are called Phosphodiester Bonds. You might want to remember that. And the differences between RNA and DNA are rather simple. First off, the nitrogen bases in DNA are adenine, thymine, guanine and cytosine, and in RNA, instead of thymine, it has uracil. And DNA is a double-stranded structure, and RNA is only a single-stranded structure. So like I told you guys, the DNA molecule was first found by James Watson and Francis Crick, and they found this structure and they named it the Watson and Crick model. It's a double helical structure because the strands of DNA keep on spinning and they go down and down and down. It consists of two polynucleotide chains which are antiparallel. Each nucleotide chain consists of three parts: a deoxyribose sugar, a nitrogenous base, a phosphate molecule. The nitrogenous bases are of two types, purines and pyrimidines, and the nucleotides are the building blocks of these polymers. Another small fact you might want to remember is that between every adenine and thymine base, there are 2 bonds, and between every guanine and cytosine base, there are 3 bonds. And these bonds happen to be hydrogen bonds. These are the bonds that break when DNA gets denatured. Now remember, unlike proteins, DNAs can get renatured. So where can you actually find DNA in the living organism? It's found in the nucleus. Now if you take a cell, we'll go into the description of the cell in the next part where we talk about organelles. If you take a cell, it has a nucleus. A nucleus is where it stores all the genetic information. The chromosomes, or the condensed molecules of DNA, can be found in the nucleus. There are also circular DNA, a form of DNA which is not as complex as DNA which makes chromosomes, but it can be found in organelles like mitochondria and chloroplast. In viruses, you can see that most of them don't have DNA, and in fact they have RNA, but some of them do have DNA. So we're going into a fun part. It's called DNA replication. How a DNA molecule creates another DNA molecule. So we know that DNA acts as the hereditary material, the most important feature of it as well. The reason that it acts as this is because it stores all the genetic material, and it is capable of self-replication. That means it's capable of forming 2 exact copies of itself by using enzymes like DNA polymerase. So we discussed that the DNA molecule is a double helical structure, so that means there are 2 strands. So the hydrogen bonds break and it divides itself into 2 separate strands. And then free nucleotides attach to their relative bases. And finally it creates half of the original material. So because of this replication, a lot of errors could occur, and that's generally how you get mutations. During replication, a nucleotide might break, it might go missing. Sometimes additional nucleotides are attached to the DNA molecules and it doesn't get the right sequence it wants, because we all know that the order in which these bases are stored is what gives you the genetic information. For example, if you say that a DNA molecule with the order of ACCCGTCGT, which gives you the color for your skin, maybe an additional nucleotide joins that, the color of your skin would not be the same because of the mutation, and that's what happens during mutations. Just like proteins, well, just like anything, DNA could also be denatured. When you bring a DNA molecule up to a near boiling point, which is around 90 degrees Celsius, the hydrogen bonds break between the 2 strands. But the greatest thing is, unlike all these proteins and other molecules that break, after the DNA strands get broken down into 2 separate nucleotide chains, they can always reconnect. The hydrogen bonds reform to recreate the strand. Now, because this process is reversible, it ensures safety, and that's why DNA molecules are a great source to store genetic information. So that's pretty much it to know about DNA. It is a very complicated subject, and we don't really need to know so much about it, but if you are really interested in learning about DNA, it's a magnificent field to go about, because everything that you need to know about DNA hasn't been found out yet, so there's always time. If you're interested in that, that would be great. So we're off to RNA. The first thing you need to remember is that a DNA is a double-stranded structure and RNA is a single-stranded structure. Therefore, RNA is a bit less complicated. But there are three types of RNAs. One is called rRNA, mRNA, and the other is called tRNA. rRNA means it's a ribosomal RNA, and it's around 80% of all RNAs. The mRNA is considered as a messenger RNA, and it's around 5%. And finally, tRNA or transfer RNA is about 15%. Ribosomal RNA, it's called ribosomal RNA because it's involved in the formation of ribosomes. Now, ribosomes are these little globules that you can find inside the cell. mRNA is a single strand which is created by the nucleus. Now, during DNA replication, the mRNA takes a copy of the DNA base sequence and it carries that to the ribosomes. So that's what the mRNA does. It generally carries genetic information copied from the DNA to the ribosomes. And a tRNA is a small molecule. The main function of tRNA is to transport amino acids to ribosomes for the synthesis of proteins. If you remember that we talked that the human body contains about 20 types of amino acids which create protein, and because of this there are different types of tRNA. So we were talking about protein synthesis for all this time and we really didn't have a clue what it was. So let's just get into a brief description of protein synthesis. It's broken down into two general parts. One is called transcription and the other one is called translation. You just need to remember that transcription means copying. So transcription is the process by which a complementary mRNA copy is made of specific region of the DNA molecule, which codes for a polypeptide. Since we're talking about DNA, you might want to know the definition of a gene. A gene is the specific region of the DNA molecule that codes for a particular polypeptide chain. So remember that again, it is a specific region of the DNA molecule that codes for a particular polypeptide. That is what a gene is called. Now, the unwinding of the DNA is the result of the hydrogen bonds between the purines and pyrimidines, which break up and it separates it into two different strands of nucleotides. So along these strands, it attaches its complementary RNA nucleotide using enzymes like RNA polymerase. A guanine base on a DNA will attract to an RNA nucleotide with a cytosine. However, you should remember that RNAs have uracil and they don't have thymine. So an adenine base of a nucleotide in DNA will attract to a uracil base in the RNA instead of thymine because the RNA does not have thymine, and vice versa. An RNA uracil can only attach to a adenine of a DNA. Once the mRNA is formed, it separates from the DNA and moves out of the cytoplasm. And then it attaches to a ribosome. The main function of the mRNA is to carry the genetic information stored on it, or the code, to form a certain polypeptide chain from DNA to the ribosome. So this process was called transcription, and then we go to the next part, which is called translation. Translation is the sequence of nucleotides in a gene determined by the sequence of amino acids in a protein. 3 adjoining nucleotides in a gene that symbolizes one amino acid in protein synthesis is termed a codon. You need to remember what a codon is. It's a sequence of 3 adjoining nucleotides that symbolizes one amino acid. So for example, you can say that AGT would make an amino acid. So that specific AGT would be called a codon. So the tRNA molecule transports amino acids to the ribosomes, and the complementary anticodons of tRNA, it's the opposite one or the complementary ones of the codon, carrying the specific amino acid, is attached to the complementary codon of the mRNA. The function of the tRNA molecule is to carry amino acids to the ribosome for protein synthesis. loads of these amino acids get together and join to form proteins. So the RNA reads the code of the mRNA and helps the formation of polypeptide chains. Assembly of polypeptides take place on ribosomes. Once the polypeptide is formed, the mRNA and tRNA are released back into the cytoplasm. Also remember that if a bunch of ribosomes are in one place, it's called a polysome. So let's see if we remember this. First, there is a DNA strand. Let's take an example of ATGGAAC. And during transcription, it breaks into the two separate strands of ATGGAAC and TACCTTG. So it joins with its relative RNA, the UACCUUG, and again AUGGAAC. So these are the complementary mRNA molecules that attached with it. And then it moves on to the ribosome, where the tRNA is used for protein synthesis. And finally, they're all released back into the cytoplasm of the cell. As we said, DNA is a molecule which is great, or which is ideal to carry genetic material, because it has the ability to store information as a sequence of bases. It has the ability for self-replication. It is a stable structure, and it is universal. Every organism can have it. And it has the ability to have mutations, because evolution can't happen if DNAs don't have the ability to mutate. Then we would completely keep our genetic material and we would just send it through generation to generation without a slight alteration. Now, don't forget that mutation is not always a bad thing. It could be a bad thing, and mutations are generally anything which changes the sequence of bases in a DNA. So again, the main function of DNA is the transmission of genetic information, and the main function of RNA is protein synthesis, but in some organisms like viruses, genetic information is also stored. There are other nucleotides instead of just DNA and RNA, even though we just said DNA and RNA to begin with. So the other nucleotide we're going to go into is adenosine triphosphate, also known as ATP. ATP is composed of a purine adenine linked to 5 carbon sugars and 3 phosphate bonds. When the bonds of the phosphate groups are broken and are hydrolyzed, it releases energy, and that's how ATP creates energy. This catabolic activity releases energy. So then you have ADP. Again, ADP can hydrolyze again to create AMP. So the main function of ATP is to store energy, and it's also used to transport phosphate groups into other molecules. Now that's called phosphorylation. The other nucleotide we're going to talk about is FAD, flavine adenine dinucleotide. Now this is a bit different because it's a dinucleotide. It has 2 flavine mononucleotides in 2 sides connected by 2 phosphate groups and a adenine on either end. The main function of FAD is to act as a hydrogen carrier. And the final nucleotide we're learning about is called NAD, Nicotinamide Adenine Dinucleotide. It's just like FAD, it's also a hydrogen carrier and a reducing agent. NADP is a hydrogen carrier in photosynthesis. You're going to learn that a lot because photosynthesis is one of the hardest but yet most interesting parts. So there you have it, guys. You don't really need to know much about FAD and NAD. The things you need to memorize here are the functions of DNA, the functions of RNA, the structure of DNA and the structure of RNA, and how protein synthesis works. If you can get all 5 of those done, you are ready to go to the next part. But since the part about codons and anticodons is a bit confusing, I know that could be really confusing at first, we'll go into like 1 or 2 questions to see if you have a basic idea of what you need to know. So we're going to ask the question based on a hypothetical strand of DNA. So let's take the code ATTCGA. Okay, we got ATTCGA. So for this code, what is the base sequence of the other DNA strand? So We studied that the purines have the opposite pyrimidines. A has T and G has C. So you need to basically, if there's an A, you write T. If there's a G, you write C. If there's a C, you write G. So ATTCGA would be TAAGCT. So that is how you would get that. And then what is the base sequence of the mRNA strand? So now we're asking the mRNA strand of the first DNA strand we mentioned, not the opposite strand of that. So what is the mRNA strand of ATTCGA? Now you remember that adenine would bond with a thymine in a DNA molecule, but in the RNA molecule there is no thymine. So instead of T you write U. So it would be U, and for T it would be A, G, C, and U. And there are some neat other questions that they can ask. For example, let's say that adenine is 40% of nitrogenous bases of a DNA strand. What percent of guanine can you find? So you know that the amount of adenine and the amount of thymine is the same. So if it's 40%, you know that there is 40% thymine. So that would end up to a total of 80%. And you would also know that the total of guanine and cytosine would be 20%, since 100 minus 80 equals 20. And then you can say that the amount of guanine is equal to the amount of cytosine you have, and you can say that the amount of guanine in the DNA strand is 10%. So just like that, you need to be able to make some simple calculations. And let's just get into the very last part about the codons and anticodons. So if an anticodon of a particular amino acid in a tRNA is AAA, what is the DNA code for the particular acid? So in a tRNA is AAA. So you know that tRNA is coded from an mRNA strand, and that would mean it would be UUU. Since RNA there is no thymine, it would be UUU. And the mRNA is coded again from the DNA strand in the very beginning, and that would also be AAA. So you would realize that the anticodon of the tRNA would be the same as the DNA. So if you understand that, you can pretty much go through this lesson. That is all you need to learn about nucleic acids. Let's just wrap it up now. It was a rather long episode because nucleic acids are long, literally, because DNA strands have hundreds of thousands of bases and they form these massive chromosomes, and it's beautiful. So we will see you guys next time. I hope these videos are helping you guys out. Again, if you guys have any questions, leave a comment below. We will try to get that answered. And we're also working on a forum for the website. We'll keep you posted about how that goes. If you want to visit the website, the link is www.LankanTutor.com. Again, thank you guys for watching. We will see you guys next time. As always, have a great day. And don't forget to like, comment, share, and subscribe.